Bile is a yellow-green substance that we have probably all encountered on an unfortunate day when we happen to be sick and our nausea progressed to vomiting. That yellow-green substance that we see that's very bitter is bile. And what it does is its job is to coat a lipid so that the lipid can actually bind to an enzyme. We have said many times in our digestive lecture that lipids, being fats, cannot have enzymes called lipases to bind to them that should break them down because the enzymes are water-based and the lipid repels them because it is a lipid. It does not interact with water. So what a bile molecule does, specifically a bile salt, is it has two parts. I have sketched this on this particular slide. This is a creation that I made that shows a bile salt. Part of the bile salt is what we call hydrophobic, meaning that it binds to a lipid. The other part of a bile salt is hydrophilic, which means that it can interact with water. So you'll notice that the bile is going to have these salts attached to a lipid with the hydrophobic part touching the lipid, two lipid parts together, and the hydrophilic part being exposed to the outside environment, which typically is aqueous or water-based. And that would then allow the ability for a lipase, which is water-based, to bind to a lipid and ultimately break it down. So that's the importance of bile. So bile is going to coat lipids for enzyme attachment. That's its purpose. And bile is composed of many things. It has cholesterol as part of its components. It has neutral fats. It contains phospholipids. It contains various minerals and these really important bile pigments that we were just observing. And these I said that wrong. Bile pigments are going to ultimately be used to give the color to feces. The bile salts, which is the last dash there that my eye was looking at as I was saying pigments, sorry there. The bile salts are going to help us digest fats or break down fats. And these bile salts that are so important for being able to allow the enzyme to bind to the lipid are actually going to be taken back up in the last part of the small intestine. They're going to be reabsorbed by our ileum so that they are not lost as a waste product. They're not going to go out as part of feces. They will actually stay in the body. Now, when bile is made, it is going to be released out to the bile ducts and it will leave the liver out of what are known as the hepatic ducts. So our liver would sit roughly right here and on this diagram, and coming from the liver we would have bile collected by all those little bile ductules surrounding the hepatic lobules, and those little ductules all merge together into these main right and left hepatic ducts if we are facing someone. And they are going to fill with bile, and as bile fills these little tubes, it will ultimately fill this tube, which is known as the common bile duct, starting about right here, and we'll talk about why we start it right there specifically. This is known as the common hepatic duct, where the right and left hepatic ducts merge. We're not going to get quite that fancy. We'll just talk about right and left hepatic ducts, or just hepatic ducts coming from the liver. Now, there's another little tube that branches off the side of this common bile duct that's going to come down here and ultimately empty into the duodenum, or the first part of the small intestine. And that extra little tube is this one right, wrong color, this one right here, and it is called the cystic duct, and it leads over into the gallbladder. So what's going to happen is, as this liver, where I drew this larger green outline, makes bile and directs that bile into, I'm going to go with yellow here, into these main hepatic ducts here and here, and we fill the last of the hepatic ducts. Then the bile is going to move down into this common bile duct, fill it, and if this little sphincter that enters into the duodenum is closed because there's no food coming into the duodenum at this point in time, any extra bile, because this liver makes bile 24/7, 365 days of the year, 366 if we have a leap year, we are going to have a place for that bile to go. And what happens is when we fill all these tubes where we have the yellow, then extra bile that is being made is going to back up into this tube, which is the cystic duct, and be stored in this little sac. And this little sac is called a gallbladder. And so the gallbladder's job is just to hold extra bile that was made by the liver until it's needed. So if digestion's not occurring, there is the little sphincter that we learned in lab. It's called the hepatopancreatic sphincter. And that little sphincter, let's see if we can clear off a little bit of all this, oh goodness, excess jotting. Let's see. We can pull that off. So we should, in theory, be able to take away, there we go, all this extra that we had going there. So our hepatopancreatic sphincter would actually sit right here, roughly. If I can get the pen to work again, let's try this button. There we go. So it sits right here. And this little sphincter, if it is closed, would prevent bile from entering into the duodenum. So as it comes from the liver, I'm just going to pick the left hepatic duct a minute, and fills the common bile duct, any extra would go over into the cystic duct and be stored in the gallbladder. However, when food is present, then that would change. We would have that little sphincter to open, and all this bile that has been awaiting entering into the duodenum would have the ability to start moving in. And with food presence, we would actually want this gallbladder to contract and squeeze extra bile that's already been produced out into that common bile duct so that it also can make its way down into the duodenum. So as we look back at our slide information, it mentions that without digestion, the hepatopancreatic sphincter is closed and bile is going to be stored in the gallbladder. So the route of bile production would be bile would enter the hepatic ducts, and then that bile would go into the common bile duct. And once it's filled, then the bile would enter the cystic duct to be stored in the gallbladder. And there should be an R on that gallbladder. Sorry, I ran out of room there. And that's the route we've been looking at for the last few minutes. Now, if chyme, foodstuffs containing lipids or fats, comes into the duodenum, then a hormone is going to be released called cholecystokinin, or abbreviated CCK, once we write the word out one time. And once CCK is released, we get some extra assistance with moving bile into the duodenum with the presence of lipids to be broken down. As I mentioned a moment ago when we finished looking at our diagram, the gallbladder would contract. We will also see pancreatic juice released into the duodenum, and we would have the hepatopancreatic sphincter open to allow the gallbladder to release bile into the duodenum, and the pancreatic juice to release its collection of enzymes for breaking down foodstuffs as well. So if we slide down here, what would occur is with excuse me, with cholecystokinin secretion, that would cause bile to be released from the gallbladder by way of the gallbladder contracting. that would have bile come from the gallbladder up the cystic duct into this common bile duct, assuming the hepatopancreatic sphincter or ampulla levator is open, that bile would go into the duodenum. And since the liver is always making bile, any new bile coming from the hepatic ducts would also enter into the bile going into the duodenum. As that's occurring, the opening of that hepatopancreatic sphincter also allows these ducts from our pancreas that we looked at in lab to allow pancreatic juice to enter in that hepatopancreatic sphincter also. It will include a lot of enzymes. It's also going to include bicarbonate to neutralize the acidity of the chyme coming from the stomach. So, as you might expect, our next stop is going to be a look at the pancreas. Let's take a look at hydrochloric acid production at the stomach and the creation of the alkaline tide on this little diagram that's just a simple sketch. So first off, let's orient ourselves so that we can know where exactly we are. So this is our stomach. We would have, of course, our esophagus coming in here, and the little dot that you see here is the pyloric sphincter. Now, that's going to take us on around to, this is going to be the duodenum, the first part of the small intestine. And we have a little inset here that is a somewhat oddly shaped structure. This is supposed to be our pancreas. And far over here to the edge, we have a little blood vessel. An artery. Now, before we stop our orientation, let me, and I have zoomed this in just a little bit for observation of this little section here right around the stomach and this vessel. We are going to, on our stomach model, call this little corner over here a really big parietal cell. So obviously this is a hugely, grossly large parietal cell, but it's for the purpose of allowing us to draw in our carbonic acid bicarbonate equation. So let's take a look at what it is that occurs for the production of hydrochloric acid. So if we consider one of the functions of the parietal cell is to make hydrochloric acid, as we just looked at the diagram in your book, we would have carbon dioxide, which would be in the parietal cell from functions that the cell carries out on a normal basis, like making ATP, and water, which is, of course, part of the cell's cytoplasm. Those two would come together and make H2CO3, carbonic acid. That carbonic acid would then break down into hydrogen ions and bicarbonate. The hydrogen ions are going to move over into the middle of the stomach where foodstuffs are coming in to make the environment of the stomach more acidic. As that hydrogen ion moves over, we're also going to move this bicarbonate into the blood. The presence of that bicarbonate in the blood means that we can bind to more hydrogen ions in the bloodstream, which means that bicarbonate actually is acting as a base. And that's going to make the blood slightly basic, which we call the alkaline tide. Now, this is not going to last very long. We're going to reverse this as the blood passes by the pancreas, and the blood is moving in this direction where the red arrows are going. So this is not going to last for an extremely long time. But there is an advantage to this alkaline tide that is occurring, and the advantage that we're going to find is this unique little piece. The alkaline tide's created by this negatively charged bicarbonate moving into the blood, and to also keep the charge of the blood from changing and becoming more negative in addition to becoming more basic, we're going to swap a chloride ion for that bicarbonate ion, and it's going to be allowed to pass through that parietal cell and enter into the stomach. And of course, it could combine with that hydrogen ion to make hydrochloric acid, which is actually what we're after. But as we said a few minutes ago, we are just as likely to find that hydrochloric acid in the form of hydrogen ions and separate chloride ions as we are to find it as a whole structure, HCl. So what we'll do is in the next few minutes, we'll take a look at the rest of the information on the stomach, and then we'll migrate our way into the small intestine. And as we do so, we'll talk about the events that occur in the first part of the small intestine called the duodenum. And one of those events is the production of some hydrochloric acid. I said that wrong, is the production of some bicarbonate that will be moved over into the duodenum to neutralize the hydrochloric acid coming from the stomach. And in that process, we're going to neutralize the alkaline tide in the blood and return the blood back to its normal pH. So we'll come back to this diagram, just set it aside, and before we begin the large intestine, we will have completed a little bit more on this picture. One function of the parietal cells is the production of stomach acid, known as hydrochloric acid. This is created by utilization of an equation known as the carbonic acid bicarbonate equation, which can utilize an enzyme called carbonic anhydrase to speed up the chemical reaction. However, this enzyme is not required during this reaction. If we look at the carbonic acid bicarbonate equation, we need to first realize that this equation can run in two directions. So I have shown it running to the right on the page as it's printed, but the equation can also run to the left. The direction the equation will continue depends upon how much of each of the substances on the far right and the far left are present. So if we have more CO2 in water, then the reaction is going to run to the right as the equation is printed here. If we have more of these two substances on the end, called bicarbonate and hydrogen ion, then the equation will run to the left. So let's introduce the components that are part of this equation. So first off, we have carbon dioxide, or CO2, joining with water. When carbon dioxide joins with water, we form an acid called carbonic acid, which is the middle chemical. This carbonic acid is known as a weak acid, which means that it's going to break down slowly over time. As it does so, it forms the molecule bicarbonate, which is the way our bodies essentially hide carbon dioxide in our bloodstream. And that bicarbonate is HCO3 with a negative charge. In addition to bicarbonate, carbonic acid breaks down to release hydrogen ions all by themselves, which means that our solution is going to become acidic. Because anytime we have hydrogen ions, or H+, all by itself, not attached to anything else, we have a more acidic environment. So with the production of hydrogen ions in the parietal cells, what happens is these hydrogen ions are moved from these little gastric glands into the middle of the stomach, known as the lumen, and there it's going to combine with a chloride ion to form the hydrochloric acid. The bicarbonate that's produced as part of this equation is not something we really want right here. We made it simply to get those hydrogen ions. So that bicarbonate is going to be pushed out of the parietal cell over into the bloodstream, where we're going to actually swap it for something we do want here, which is a chloride ion, which will help us make our hydrochloric acid in the stomach. And as we move that bicarbonate over into the bloodstream, that's going to make the bloodstream a little bit basic as far as its pH, which creates something called the alkaline tide. So this is going to be the addition of HCO3 minus to the blood. And that's going to create a basic, an alkaline environment. Let's see if we can make that just a wee bit prettier. How about that? A little bit better. All right, so let's mention what it is that hydrochloric acid is actually going to do in the stomach once it is produced. So the purpose of hydrochloric acid is actually numerous. One thing that hydrochloric acid does, and the function that we have it for here in the stomach in the digestive system, is it's going to activate enzymes. The primary enzyme we're interested in activating in the stomach is pepsinogen, which is going to turn into its active form of pepsin, which will help us to break down proteins into smaller amino acid chains. An additional thing we'll find happening here in this process that we've discussed to date is we will find that hydrochloric acid will actually have the opportunity to activate lingual lipase that we made in the saliva. However, this enzyme, as we previously mentioned, is essentially not going to do anything because even though we've activated the enzyme can't bind to its target, which is the lipid, to be broken down because they are simply different kinds of molecules. Lingual lipase actually interacts with a watery environment, and lipids are not any part of a watery environment. They are the antithesis of that. And so the lingual lipase enzyme actually is rather a dud. It's really not going to do anything. A second function of hydrochloric acid, as is listed here, is that hydrochloric acid does help us to break down plant materials that we consume, so things like lettuce or broccoli or other such foods that our bodies do not possess enzymes to actually break down. So it'll help to break down some of the fibers, soften it a little bit, make it a little bit easier to move throughout the digestive tract. A third function of hydrochloric acid is for utilization of iron. So one thing hydrochloric acid does is it changes iron into a usable form from Fe3 to Fe2, from ferric iron, Fe3, to ferrous iron, Fe2. And Fe2 is used for production of hemoglobin, which carries oxygen in our bloodstream so that we have oxygen around the body. And the fourth and last thing that is mentioned here as a function of hydrochloric acid is something that we have mentioned for some time, which is that it breaks down bacteria, viruses, foreign bodies that happen to come in within food and drink. And that's certainly an advantageous function of this stomach acid. So if we look, and I'm sorry, I keep trying to scroll my page and write instead. As I scroll my page here just a moment and look at the picture from your text, we are looking at a little red parietal cell, as it is labeled, as part of the gastric gland. This is the inside of the stomach where the food would be passing through. We would have our chyme. And what's going to happen is we're going to carry out this carbonic acid bicarbonate equation. So as you can see here, CO2 binds with water, a little bit small, see if we can zoom in a smidge, and that's going to form carbonic acid. And carbonic acid is going to break down into hydrogen ions, which are going to be moved into the lumen of the stomach, going through a little protein here. It's usually swapped for potassium. And then we're also going to break down carbonic acid into bicarbonate, and bicarbonate is going to be moved over, as we see here, into the bloodstream. As a negative ion, it's going to be swapped for chloride, which is also going to be moved into the stomach lumen. And when we get hydrogen ion and chloride ion together, we're going to make hydrochloric acid. But we do need to be aware that even in the stomach lumen, hydrochloric acid is usually not found in the form I just circled. It's not formed with H and Cl together. Instead, these ions quite frequently are separate because the advantage of that is, as we said a little bit ago, anytime we have hydrogen ions by themselves, they make an environment acidic. If I bind them to something, even something like Cl, that is going to reduce their effectiveness. They're going to not make a particular solution, a particular environment quite as acidic. So if you will, grab your picture that was posted as an item to print, the stomach picture, and we'll come back in the next video and take a look at this concept of production of hydrochloric acid for the stomach and creation of the alkaline tide on a picture that we have on our own, and then we'll use it a little bit later to reverse that alkaline tide as we move foodstuffs closer past the pancreas and the duodenum. Be right back. As food exits the stomach, it will enter into the small intestine as the next place along the alimentary canal. Soon after entry into the small intestine, however, we will have function of several accessory organs, the liver, the gallbladder, and the pancreas that will play a large role in digestion. So letter E takes us from the mouth to the pharynx, to the esophagus, to the stomach, to now the fifth location along the alimentary canal, the small intestine. And the small intestine extends from the pyloric sphincter, the exit of the stomach, the entry to the small intestine or the duodenum, all the way down to the end of the small intestine where it joins with the colon, which is known as the ileocecal valve. It can also be called the ileocolic sphincter. There we go. There are three regions to the small intestine. It is roughly about 20 feet long in an adult. The first 10 inches of the small intestine wrap in a C shape around the edge of the pancreas as the small intestine begins to form past the stomach. That region is known as the duodenum or the duodenum. Following the duodenum is the jejunum, and it's roughly about 8 feet in length. And the end of the small intestine is the ileum, which is 12 feet in length. By far and away, the duodenum is the place where most activity occurs. That's where we have the capacity to break down any type of macromolecule due to enzymes coming from the pancreas. The duodenum, therefore, being the place where most substances are broken down in the body, is also the place where most things are absorbed. The jejunum, about its first third to half, will allow still some absorption of substances, nutrients that have been broken down. Past that point, anything else is just movement of food forward through these tubes. In the small intestine, there are some features that assist with absorption, especially in the duodenum and the early part of the jejunum. Those include the folds that we see on our models in lab in the duodenal openings called the placae circularis. These are ridges of tissue which allow more absorption to occur. And along that placae circularis and throughout the duodenum and a little bit of the jejunum, we also have folds of the mucosa known as villi. And then those little folds have additional folds, which are called microvilli. And that makes sure that there is more than ample space for absorption of nutrients to occur so that we don't have any loss of potential nutrients of food passing through the intestines. Then we need to mention that there are two ways that we move things in the small intestine. One is segmentation and the other is peristalsis. Segmentation has the specific goal of mixing, especially the pancreatic juice early in the duodenum, with the broken down food particles to make sure that we, to the best of the ability of the digestive system, mix up all the foodstuffs with all the potential enzymes so that everything can be broken down really well. Segmentation is a bit of a unique movement that is carried out solely by the activity of the small intestine. And what it does is it mixes slowly food substances. And I was thinking that I had a diagram right here. I believe actually we're going to find it in the very first section of PowerPoint set 2. But we do have a segmentation diagram where it shows how we can mix the substances in the small intestine for better absorption. And then towards the middle of the jejunum and through the ileum, we have just simply movement of substances through the tube. And that's just to get the substances moving towards the large intestine for removal from the body, for waste removal. And that's just normal peristalsis, normal movement of substances through the tube by way of progressive contractions of the circular and longitudinal layers of smooth muscle. Now, the wall of the small intestine has all four tunics, the mucosal layer, the submucosal layer, the muscularis layer and the adventitial layer. The uniquenesses that we find in the small intestine is that the mucosa has microvilli for extra absorption, especially in the region of the duodenum and the jejunum. We have good numbers of goblet cells to be able to adhere to the particles that are being broken down so that we can absorb them well. We also have the ability to secrete what's known as intestinal juice, which includes things like lysozyme, an enzyme to break down bacteria. And we have enzymes that are attached along, especially the duodenum and the first part of the jejunum, to the wall of the mucosa. And those enzymes are called brush border enzymes, and they're going to activate inactive proteins that are being secreted by structures like our pancreas. Then we have in the submucosa some unique substances early and also late in the small intestine. Early in the small intestine, we see duodenal glands, or what we call Brunner's glands, which secrete mucus, and their job is to protect the inner lining of the duodenum from the acid of the stomach, the chyme that's coming in. Then later in the small intestine, in the submucosal layer, we have Peyer's patches. These are structures that are going to help to break down bacteria. And they're present because the large intestine has its own bacterial cluster. It has its own bacteria for the purpose of breaking down vegetation that we consume that our bodies don't make enzymes to break down. So those bacteria that are in the large intestine that should not be in the small intestine, but unfortunately will come visit when the ileocolic sphincter opens, the Peyer's patches help to get rid of. So we will talk about in more detail some of these structures of the small intestine as we move forward. So these are some diagrams from your text that show the duodenum here wrapping around the small intestine. Then we have a look at the jejunum that would follow that duodenum in the upper portion of the small intestine, and the ileum, which would usually be the lower portion of the small intestine. We also have a look at some of the wall of our small intestine so that we can see some of its features. We have a villus, a finger-like extension here that we can see, a fold. We also see the goblet cells that secrete mucus. We also have the Paneth cells that secrete the lysozyme to break down the bacteria. We have goblet cells for additional mucus secretion that are shown a little bit smaller in this side. And we will talk about some structures later like the lacteal, which can absorb some of the larger substances that our digestive system will help us to break down. So we'll look a little bit more at some additional small intestinal components in a bit. So the stomach functions by way of utilizing 3 different phases. Initially, the stomach begins its activity before food ever reaches it by simply smelling, seeing, or having us think about food. In those instances, our medulla of our brainstem will send a signal to our vagus nerve to go ahead and begin to produce the gastric juice. When food actually reaches the stomach, that begins a second phase of gastric function, and several things occur simultaneously as food enters into the stomach. First up, just simply the presence of food stretching the stomach causes acetylcholine release, which triggers parietal cells to secrete hydrochloric acid. Secondarily, most of the time food is going to going to actually have within it some portion of protein. The protein presence will increase the pH of the stomach. It reduces the acidity, in other words, and that's going to cause additional hydrochloric acid secretion from the parietal cells, often assisted by G cells of the gastric gland secreting gastrin, which further stimulates the parietal cells to secrete hydrochloric acid. That hydrochloric acid then, that has been secreted due to protein presence and an increased pH, will act to break down pepsinogen, and it also will act to make hydrochloric acid new from the parietal cells. Then we have histamine from histaminocytes when we have food present, and that also activates the parietal cells to cause hydrochloric acid secretion. So the gastric phase actually produces about 2/3 of gastric juice. So by far and away, the majority of the gastric juice is produced during this phase as food enters the stomach. Then, as we have food begin to move out of the stomach, that's going to stimulate the intestinal phase to begin. And what happens there is we have intestinal gastrin secreted as the chyme moves into the first portion of the small intestine called the duodenum. That's going to cause what's known as the enterogastric reflex, which causes the stomach secretions to begin to end because we have food now in the small intestine and we're assuming that our meal will be ending soon. And that is going to then allow the chyme to go into the duodenum and trigger the secretion of secretin, cholecystokinin, and gastric inhibitory peptide, all of which will cause gastric inhibition and the closure of the pyloric sphincter. Food moves in the stomach by way of a process called peristalsis. So what occurs is as the peristaltic waves of the esophagus allow food to enter into the stomach, that's known as receptive relaxation. So the cardiac sphincter opens and the stomach is allowed to take in a bolus, and then it goes through a process called adaptive relaxation where we take that bolus and expose it to the product of the gastric glands or the gastric juice. That's going to produce a good little volume in the stomach. So when we get to about 1 liter of food in the stomach, we have some pressure in the stomach that actually triggers peristalsis at the cardiac sphincter and moves the food down toward the pylorus where we have thicker muscle, and we grind and churn the food more and break it down even better. And the very end of our pylorus is a small region that holds about 30 milliliters of chyme, and that little region is known as the antrum. And please do fix this slide. Every time it was typed, for some reason, it wanted to call it chyme instead of chyme, but that should be CHYME. Antrum holds about 30 milliliters of chyme. Now, as we move foodstuffs down into that region that is known as the antrum, we are going to use peristaltic waves. And it's very important that we don't have a whole lot of this chyme entering into the duodenum, the first part of the small intestine, all at once, because it's exceedingly acidic and it could bore through the tissues that make up the small intestine and essentially disintegrate the entry into the small intestine. That would not be good. This chyme also is very nutrient rich, which means it draws water to itself, which would be dehydrating over a period of time. So we only allow little bits of chyme to move from the stomach into the small intestine at one time, and that's going to allow reduction of the number of nutrients that would draw water out from cells in the abdominal cavity, and it's also going to neutralize that chyme and make it less acidic. So the way that we control food substances leaving the stomach is by way of a group of little cells called these cells of Cajal. And these are self-depolarizing cells that automatically function, and they are going to send 3 milliliters of chyme 3 times a minute into the duodenum. So that's 9 milliliters of chyme in a minute's time to give ample time to reduce the acidity of the chyme and also reduce its heavy content of nutrients. Now, how long does it take to digest a meal and move food out of the stomach? Well, on average, it takes about 4 hours. And it takes a little longer if you've got a lot of fat that you consume in your meal. And large meals or meals that are liquid actually digest a little bit faster. So let's take a quick peek at intrinsic factor. Is the only substance that the stomach is really needed for. Intrinsic factor is secreted by our parietal cells alongside hydrochloric acid, but this particular substance is used for vitamin B12 absorption in the small intestine. Intrinsic factor must be supplied if someone has their stomach removed. So a person can live a perfectly healthy life without a stomach. They just need injections periodically of intrinsic factor. Then our chief cells create a different chemical called pepsinogen. That was the zymogen, the inactive protein that when it's activated by hydrochloric acid becomes pepsin, which is going to break down proteins into smaller pieces to begin their initial breakdown. So the mouth is the site of carbohydrate breakdown, starting with breaking down starches by salivary amylase, and proteins begin their breakdown in the stomach with activation of pepsin. So this little diagram is one that I like from a previous McGraw-Hill text that shows how we activate pepsinogen and where pepsinogen and hydrochloric acid actually originate from. So this shows a little piece of protein that is coming into the stomach, and we have coming from the chief cells some pepsinogen in the lumen of the stomach, coming from the parietal cells some hydrochloric acid, and that hydrochloric acid is going to break down this pepsinogen to remove this little yellow portion of the pepsinogen and leave the active form of the enzyme called pepsin. Now, when pepsin is exposed to proteins in food, it is going to break those little proteins down into smaller pieces, sometimes by random chance, single amino acids, sometimes pairs, sometimes fours, sometimes fives, sometimes eights, but we'll break the amino acid chain into much smaller pieces so that they can then be broken down as we continue through the small intestine. Then this is your book's rendition of the exact same thing. We have chief cells that are purple, parietal cells that are red, and we are looking here deep down in the gastric pit depression at the bottom of the gastric gland. And we have our parietal cell here in purple that is secreting out some hydrochloric acid. I said that wrong. The chief cell's in purple, and it's secreting out this pepsinogen. I was thinking that the diagram had an error on it. The parietal cell is labeled way over there on the left, but it's pointing to this little cell right here that's red. So this little parietal cell is correct in that it makes hydrochloric acid, and that hydrochloric acid comes into contact with that inactive pepsinogen, known as a zymogen, activates it, turns it into pepsin, and that allows the proteins that are present in foodstuffs in the stomach to be broken down into smaller little pieces for absorption. So let's take a quick break and we'll come back and look at gastric motility. The purpose of the stomach is to begin protein breakdown. In this process, food is changed from the bolus, which is a solid mass, into a liquid slurry, and that is called chyme. The stomach has the capacity, when it's empty, to hold about 50 milliliters of fluid, which is not very much. Roughly a small bathroom cup, like you might rinse your mouth out with after brushing your teeth in the evening. And it can distend or stretch to hold upwards of 3 to 4 liters of food substances once you visit something like the buffet. Now, our stomach wall is going to be composed of numerous different cell types with various different functions. Our stomach wall has an inner layer known as the mucosa layer, just like any other hollow tube in the body. So the stomach is going to have the tunics that we spoke about in the esophagus. We'll have the inner mucosa. We'll have a submucosa layer that has more glands than anything else. Then we'll have a muscularis layer, but the stomach's muscularis is a little different. And then the external adventitia layer. Now, if we look here at the stomach wall, I have just addressed of those 4 layers that the stomach wall possesses what is unique in the stomach. So the mucosa layer is very unique in the stomach. It is a columnar epithelium. That is because columnar tissue has the ability to secrete and also to reabsorb substances, and that's an important thing for the stomach to do. And the stomach mucosa actually has some deep depressions in this tissue where there are special kinds of cells within these little regions known as gastric glands. So if we take a peek at the structure of the stomach wall, we have this nice mucosal layer that sits right on the outside. So let's see if we can grab us a mucosal layer here. That's the mucosal layer there, all that little bracketed area. And we can see within that we have these little depressions that come down into the mucosa. That's known as the gastric pit. And then this region that sits even lower than that with the multicolored cells, that is what's known as the gastric gland. And we can see on our diagram here, we've got cells that are pink. We've got cells like these. We've got cells that are red like this. We've got cells that are blue. We've got cells that are green. So we've got lots of different kinds of cells in these gastric glands that sit at the bottoms of these gastric pits. And what we need to take a look at is some of those cell types. So in looking at some of those cell types in the gastric glands, we have mucus cells, and of course they're going to produce, or we'll just say yield mucus by way of the cells producing mucin that mixes with water. We also have a new kind of cell called a parietal cell. These cells are responsible for making the acid of the stomach, a special acid known as hydrochloric acid. And they also make the only chemical that we actually need a stomach for, the actual only thing we need a stomach for, which is this substance called intrinsic factor. And it's going to help us to absorb vitamin B12. Then we also have a new kind of cell called a chief cell. Chief cells release a chemical called pepsinogen. Pepsinogen is a protein, actually an enzyme precursor. So inactive proteins that are made in various places in the body are called zymogens. And pepsinogen is one of these inactive proteins. When it's first made, it's not functional. It has to be activated by another chemical. So it's called a zymogen. And then we also have down in the gastric glands regenerative cells, cells that make new cells. So these regenerative cells would have the ability to make new gastric gland cells that could become mucus cells, might become parietal cells, could develop into chief cells to replenish any cells that become old or worn or potentially damaged. Now, the stomach wall also has a unique muscularis layer. And the reason the muscularis layer is unique in the stomach is that it has not 2 but 3 layers. So most tubes just have a 2-layer muscle layer. And in the stomach, we do have the traditional 2, the outer longitudinal layer, the inner circular layer, and we have an even deeper 3rd layer, an oblique layer that lies at an angle to the circular and longitudinal layers. So these 3 layers of the stomach allow the stomach to better mix the stomach's gastric juice along with the food substances and better break that food substance down. So these gastric glands that we have been taking a look at with these various different cells, they actually can produce 2 to 3 liters of gastric juice per day. So 2 to 3 liters of hydrochloric acid fluid with proteins, enzymes ready to be utilized to break down protein, mucus, etc. There is, I will mention, a nice little inset that shows these various different cell types. So in your textbook, the mucus cells are the pink cells. So you'll notice there are a good many of those near the gastric pit. So as the mucus would be secreted going up into the middle of the stomach where there is a higher concentration of the acid. Parietal cells that make the stomach acid are actually red on this diagram. The blue or purplish cells are the chief cells that make the pepsinogen, the inactive enzyme to break protein down. And then the G cells are one of the enteroendocrine cells. They're going to release a hormone called gastrin. The stomach has actually 4 regions. One was accidentally left off of this particular slide. The stomach has regions that are somewhat analogous to a term, at least to begin with our uterus. So we said with our uterus in the female system, where the fallopian tubes connected to the uterus, there was a bump of tissue that belonged to the uterus above the fallopian tube entry, and that region was known as the fundus. This is true also with the stomach, but the entry point for one single tube is the cardiac sphincter. So it's where the esophagus comes into the stomach. We have a bump above the esophageal entry. Then we have a number 2, we'll call this 3 and this 4, a cardiac region of the stomach. And this is where food enters from the esophagus. Then we have the main region of the stomach, which is known as the body of the stomach. And then where the stomach narrows, and it also is much thicker with muscle tissue near the end, that is the region known as the pylorus. So let's take a look at those 4 regions actually on our diagram. So this is a look at the stomach, and we can see the esophageal entry here. So the region above that would be the fundus. I'm just going to label it with the letter F. Then where the food actually enters into the stomach, sort of due to gravity, that would be the cardiac region. Then prior to the stomach narrowing, this region would be the body, which is usually the larger portion of the stomach. And then the little region here where the stomach narrows is known as the pylorus. Now, the actual entry to the stomach is going to be a ring of muscle that would sit right here, which is known as the cardiac sphincter. And we can see even better here the pyloric sphincter at the end of the stomach. So we have a definite beginning and a definite end to the stomach. And then where the pyloric sphincter lies on the other side of that, we have what's known as the duodenum or the duodenum. And that would be this C-shaped region of the small intestine. And it is going to actually curve around. The pancreas is going to sit right here. And we'll see it in future diagrams. So those are the regions of our stomach. Then let's go back and finish this initial slide from the stomach. It does mention the cardiac sphincter, which is also known as the lower esophageal sphincter. So it is the sphincter or the ring of muscle tissue that closes to keep stomach acid from going up into the esophagus. But it must open to allow foodstuffs to come from the esophagus into the stomach. The pyloric sphincter is on the opposite end of the stomach towards the small intestine, and it is going to open to allow foodstuffs to leave the stomach and enter into the small intestine. Now, this food's going to be really acidic. It's going to have a lot of nutrients in it. So there are some considerations that we have to utilize just a little small opening of this sphincter periodically so that we can handle those unique chemical contents of the stomach. Then one last little set of features on the stomach itself anatomically are the greater and lesser curvatures. So if we come back over to our stomach, the wide edge of the stomach is known as the greater curvature. So this edge here, and the narrowed edge of the stomach towards the heart upward towards the superior structures of the body, that's the lesser curvature. So greater and lesser curvature, anatomical features of the stomach. Then let's quickly talk about the process of swallowing, which is anatomically known as deglutition. So swallowing has two phases. It has the buccal phase and it has the pharyngeal esophageal phase. So as we begin to swallow, our tongue is going to move up against the hard palate and it pushes the bolus up. Then we're going to see closure of structures that will help to move the bolus in the right direction. So from pushing the bolus upward, we're going to make a few anatomical changes around the mouth where we're going to have the tongue to block the mouth, the oral cavity, to move the bolus out and not allow anything to come back into the mouth. The soft palate, specifically the uvula, the very tip end of the soft palate, is going to move up to block the nasopharynx so that any food or drink that we are attempting to swallow will not go up the pharynx instead of down the pharynx as we wish it to move, or quote unquote the wrong way. We don't want it to go the wrong way. The soft palate helps us there. And then the larynx will be closed when the tongue pushes down on the esophagus. And that's going to close the larynx to move the food into the esophagus. Then once that bolus has been moved out of the mouth, the pharyngeal esophageal phase begins. And it is involuntary. We have absolutely no control over it. It is controlled by our brainstem, by the medulla and the pons regions of the brainstem, controlling muscle contractions to move the bolus or the foodstuffs down towards the esophagus and through the esophagus ultimately to the stomach. And what's going to happen is the bolus is going to move simply by peristalsis along the esophagus once it reaches the esophagus. And once the bolus gets in the esophagus, we have a reflex that's going to cause the opening of the stomach, which is known as the cardiac sphincter. So the cardiac sphincter opens to receive the food, specifically called the bolus, into the stomach. And so we can see here in diagrams from your text, figure 8, we have the preparation of the bolus. It is sitting just above the tongue, ready to be swallowed at any time. And once the swallowing process is initiated, we can see the uvula move up and back, and it closes off the nasopharynx so the food can't go up. We can see the tongue come up. We're blocking the oral cavity or the mouth, so the bolus has to go down in the direction of the arrow. And we can see that the tongue is pushing this epiglottis closed so nothing goes into the larynx there, and that's going to force this bolus down into this esophagus tube heading towards the stomach. Once we get the bolus out of the mouth, the voluntary phase, the buccal phase has ended, and the body takes over the medulla and the pons to move the bolus through the pharynx, as we see in the yellow area here, and into the esophagus. And once we get into the esophagus, it's just a matter of progressive smooth muscle contractions, aka peristalsis, moving the food into the stomach. And so this would be roughly where the cardiac sphincter sits. It's a little smooth muscle, and when the bolus begins its route down this esophagus, somewhere around at least this region, about a third of the way down, that cardiac sphincter is going to go ahead and open. It's ready to receive the bolus, and that way the stomach can go ahead and be preparing. It'll start to make gastric juice, stomach acid, in preparation for new foodstuffs entering in. So we'll take another break, come back, and pick up with the stomach. In the body, we have many hollow tubes, and tubes have some similar structures. So let's take a look at the layers of tubes, and these can also be layers of organs, like the hollow stomach. So as we look at our digestive tube specifically, we have 4 layers that we can see once we enter into the abdominal cavity. So this would apply to the esophagus below the diaphragm, the stomach. All the intestines would have these similar structures, and some features will be added or modified or tweaked just a bit for each of these different regions or organs to make them function properly. But this is the basic information for the layers of what are known as the tunics. So the 4 canal layers are known as tunics. So we have the tunica mucosa, we have the tunica submucosa, the tunica muscularis, and the tunica adventitia or the tunica serosa. You can do it either way. So the innermost layer of the tubes is known as the tunica mucosa, and at least at the esophagus, it's composed of stratified squamous epithelium. The tissue will change for the mucosa to more cuboidal and columnar tissues as we enter into the stomach, where we'll have production of mucin by those tubes to help to protect against stomach acid. We will also see that we have more of the cuboidal and columnar mucosa tissues in the small intestine for absorption and breakdown of foodstuffs that we find there as well. So the stratified squamous epithelium for the mucosa is unique to the esophagus, because we may still have some rough pieces of food that maybe in a hurry when we were eating we didn't chew very well, and they could potentially damage cuboidal or columnar epithelium that usually tends to be thinner in the esophageal location. The layer below the mucosa, so if we look at our diagram specifically of our esophagus that we left off with a moment ago, we could see, as I drew this little squiggly line in the middle of the slide, we have this stratified squamous epithelium, and I can't zoom in on it very much, but this is 20 or 30 layers of flat squamous cells. So if there is anything rough, we're not going to damage any of the blue tissue underneath, which would be considered to be the submucosal layer that's included with this larger arrow over here. And that is a good thing for the purpose of maintaining the structure and the integrity of the esophagus. So as we come down to the tunica submucosa, that's the second layer from the hollow lumen where the food passes along. The submucosa is specifically going to contain glands, and in the esophagus, we've got a lot of glands that are going to secrete mucin for mucus production to help to move the bolus down, especially if it has any rough edges to any of the food particles, with safety so that we don't have any damage to the inner tube. Now, the submucosa does have nerves that are going to extend through it, and that is known as the submucosal plexus. So these are nerve branches in the submucosal layer. Then the third layer from the inside of the tube, so lining where the food goes, we have the tunica mucosa. Then outside of that's the submucosa. Then the next layer out would be tunica muscularis. And we have two muscle layers that we find in our tubes. And we're using, of course, our esophagus as our first example to show how. We have a circular layer of smooth muscle. So it would have smooth muscle cells that go around the esophagus tube. And then we have longitudinal cells. So they're going up and down along the length of our esophagus. So we would see just cuts through the length of the smooth muscle cells on that layer. Oh goodness. So our circular layer, there we go. Our circular layer is innermost. So we could say our muscularis has two smooth muscle layers. It has an inner circular layer and an outer longitudinal layer. And together, they are going to carry out peristalsis, progressive muscle contractions to move food along hollow tubes or organs. And the muscularis layer also has a nerve collection called the myenteric plexus. So these are the nerve branches in the muscle layers and between them as well. So we said myo, mys, and sarco meant muscle. So the my, the myenteric, refers to muscle. And then we have an outermost layer farthest away from where the food's passing through the middle of the lumen of the tube, known as the tunica adventitia or the tunica serosa, and this is a serous membrane. And we said a few minutes ago that this is also called the visceral peritoneum. Now your text has an additional diagram for some tubes, and it is showing a little bit different internal anatomy for the mucosal layer. So we have the tunica mucosa. This is going to be some form of epithelium. This is showing some highly folded epithelia with some cuboidal cells so that we can have a lot more membrane, a lot more tissue, I should say, open to the environment to be able to absorb more nutrients. Then also part of the mucosal layer, if we look in a little more detail, we do have some muscle that sits just underneath that, a very, very, very thin layer. Then we have a submucosal layer, and on this particular example, it's thin, but it goes up into these little bumpy extensions that have the epithelium on the outside. That's the submucosa, and that's where we're going to again have glands that would secrete things like mucus or possibly enzymes into our tube. And then we have a muscle layer, as is labeled. That's our tunica muscularis. And then on the very, very, very outside, we've got this yellow layer. It's really thin, thinner than my little pen there. And that's going to be the tunica serosa, aka the visceral peritoneum, aka the tunica adventitia. So aka meaning also known as. So three names, same structure, just that external membrane on the outside. So a little different view, same structures, because we can have the tube change a wee bit over time. The pharynx is what we commonly call the throat. It sits just posterior to or behind the mouth, at least the 2/3 of it that we're going to use for the purpose of swallowing. So let me scoot up here just a moment. Here we go. Let's take a look at this diagram of the oral cavity. This is figure 4 from chapter 26, and it's going to show us our pharynx a bit more clearly. So the pharynx extends from where the nasal cavity narrows all the way down this tube to right here. So the pharynx itself is subdivided into 3 regions. From the narrowing of the nasal cavity down to the bottom of the uvula is the region known as the nasopharynx. Now, ideally, this is only going to allow airflow from the nose passing down towards the lungs to come through this region. Ideally speaking, from the bottom of the uvula, the very tip of the soft palate, which would be the uvula right there, to the top of the open epiglottis, this little region is what we call the oropharynx. So the region of the pharynx just posterior to the mouth or the oral cavity. And then the area where this epiglottis, this little cartilaginous flap here, closes, it moves for the purpose of closing off our airway when we swallow. So it'll move and it'll sit down this way as we swallow. And what's going to happen is the laryngopharynx is the region from the top of the open epiglottis to the closure of the epiglottis during swallowing. So that region in green is the laryngopharynx. So it's where the epiglottis moves, is the laryngopharynx area. Now, what's going to happen, we hope, in the process of swallowing is we're going to form this bolus, this mass of food that's going to sit right here. And we hope that as we swallow, this bolus is going to make its way into the oropharynx and pass down the laryngopharynx into the esophagus. That's the route we want it to take. On occasion, we have a little oopsie happen, and sometimes food will go up this way, and we call that food going the wrong way. And we'll talk about that in a moment and how that actually could happen. But that's not what's supposed to occur. We're supposed to follow the solid green line and have our bolus leave our oral cavity or our mouth, enter into the oropharynx, pass down through the laryngopharynx and into the esophagus. Now, to allow that to happen, once we get in this region of the oropharynx to the laryngopharynx, we have this tube that has as part of its structure 2 layers of smooth muscle. And those 2 smooth muscle layers, as we mentioned when we've talked about smooth muscle in the past, will progressively contract as they're stretched in a coordinated movement known as peristalsis. So this is progressive contractions of smooth muscle layers. There are 2 in all tubes, 2 layers, that is, of smooth muscle. Let me do that a little bit better. So peristalsis is the progressive contractions of smooth muscle layers to move substances through hollow tubes. It works in hollow organs too. Now we also have the addition of some skeletal muscle known as pharyngeal constrictors that lie external to the tube of the pharynx, and we looked at those in A&P 1. So if we look at the pharynx as a tube here, the red lines would include, as part of the structure of the pharynx itself, a couple of layers of smooth muscle that carry out the peristalsis. And then I'm going to use green for these pharyngeal constrictors. So pharyngeal constrictors were triangular muscles that sat outside of the pharynx to help to move food down the pharynx. There are actually 3 of these. There were a superior, a middle, and an inferior pharyngeal constrictor. And one has a little bit less of a triangular appearance, but nonetheless, roughly triangular muscles that sit in the region of the throat for movement of food down into the esophagus tube that sits inferior to the pharynx. Then we have our esophagus. Our esophagus extends from our laryngopharynx, where we have the closed epiglottis, and it goes all the way down to the opening of the stomach. So the cardiac orifice, or the cardiac sphincter, is the opening to the stomach. At the mediastinum, we have the esophagus pass through the diaphragm at a specific spot known as the esophageal hiatus. That's important. So this esophagus is going to extend from the laryngopharynx, the bottom of the laryngopharynx. It's going to go through the thoracic cavity and the region of the mediastinum between the lungs. And then it's going to pass through the esophageal hiatus. This is a hole in the diaphragm to enter into the abdominal cavity, and it'll pass from there to the opening of the stomach known as the cardiac orifice. Now, I will make a note that below the diaphragm, the esophagus gains a tissue layer. It is actually going to add a tunica serosa or tunica adventitia. So the tunica serosa or the tunica adventitia is the same thing as the visceral peritoneum. They're all the same thing. That's that outer membrane that surrounds organs in the abdominal cavity. So we have here a look at, if this is the end of the laryngopharynx, the esophagus would pass down through the thoracic cavity in the region of the mediastinum. It punctures the diaphragm at what's known as the esophageal hiatus and comes through and enters the stomach at what's known as the cardiac orifice or the cardiac sphincter, or it can also be called the inferior esophageal sphincter, all the same thing as well. So isn't anatomy fun? We have so many names for just one component. And then this is a look at a cross-section of an actual esophagus itself. So we have an outer layer, which is known as the tunica adventitia. That is only below the diaphragm, only in the abdominal cavity. Then always the esophagus has the 2 layers of smooth muscle. That's the tunica muscularis layer, as is labeled here. Then we have a layer where we have glands and some loose connective tissue. That would be the submucosal layer. And then the lining where the food passes through is stratified squamous epithelium, and that's known as the mucosal layer. So those are the layers of a typical tube in the body. So let's take a look at some digestive tubes. We'll take just a break before we come back to our tubes. Our salivary glands include 3 structures surrounding the oral cavity. The salivary glands include the larger parotid gland that we see here in front of the ear that has ducts or tubes that empty into the mouth, as well as our submandibular gland and our below the tongue sublingual gland. And each of these little glands, we'll notice, has tiny little ducts that lead into the oral cavity or the mouth. So the salivary gland is composed of two major cell types. We have the serous cells and we have the mucus cells. Serous cells secrete enzymes in a watery fluid. Mucus cells secrete, as they have from the time we first introduced them, as part of columnar epithelium, they secrete the protein mucin. And mucin is going to combine with water to, of course, make mucus for us, which will help to stick food particles together to form our solid piece that's easily swallowed called the bolus. Now, these salivary glands very obviously produce saliva, which has several different roles in the mouth. One thing that saliva does is it helps to dissolve and moisten food. That's due to the water content of saliva. Saliva also begins process of starch breakdown. So we've got carbohydrate breakdown beginning in the mouth. And this is due to an enzyme called salivary amylase. The primary enzyme that the serous cells produce as part of saliva. So amylase is the enzyme that breaks down starch. ASE ending, it's an enzyme. The other thing saliva does with its water content is it helps to cleanse the mouth. So it's going to take any foreign bodies out of the mouth, moves them towards the pharynx and the esophagus and ultimately the stomach to be broken down and destroyed by stomach acid. So this little diagram at the bottom of the picture we were looking at earlier does a nice job of showing the serous cells as well as the mucus cells of the salivary glands. And we also have a look at those mucus cells, those nice, large, plump cells that help to make mucin. So if we look at the composition of saliva, what's it actually made of? Well, it's mostly water. That's going to help deliver the components of saliva to their destination. It also contains ions or charged particles, so there would be some sodium there, there would be some chloride there, there would be some potassium there, amongst other ions. And then we have the main components of saliva that are unique to this particular fluid in the body. So the first is that specific salivary amylase. And as we said a moment ago, its job as an enzyme is to break down starch. So the main job of the mouth chemically is to break down starch. It begins starch breakdown for the body. Another enzyme is present in saliva, though it really doesn't do anything. It's called lingual lipase. It's an enzyme supposedly for lipid breakdown, but it is inactive unless it's in an acidic environment, which would mean in the case of our digestive system, this enzyme doesn't do anything until it reaches the stomach. But it has a little problem. For a lipid to be broken down, an enzyme must do the breakdown, but enzymes are water-based. And lipids are fats. And we all know that fats and water don't mix. So lingual lipase, even if it's activated by stomach acid, does not have the ability to bind to a lipid. So lingual lipase does not do anything in our body appreciably for breaking down lipids. we'll find as we move into our small intestine that there is a chemical that the liver produces that allows enzymes to bind to lipids to break them down. And that substance is called bile. And the reason bile works is that bile has two parts. It has a part that binds to the watery-based enzyme, and it has a second part that binds to the lipid or the fat-based fat that we want to break down. And I have to have that intermediary. So there are other lipases. We'll find there's a pancreatic lipase, and it will actually really break down fats later on in the digestive tract when we get down into the intestine. So lingual lipase is a bit of a dud. It really doesn't do anything even once it's activated by stomach acid. Another component of saliva is lysozyme. This is an enzyme that breaks down bacteria. And the goal of lysozyme is to remove bacteria from foodstuffs. So any bacteria that we consume will be directed towards the stomach as part of the digestive tract and would be exposed to stomach acid. But if we were to happen to ingest a large component of bacteria, a large what we call bolus, a big amount, then lysozyme can reduce the amount of bacteria before the bacteria really enter into the body very far. So we'll reduce bacterial numbers at the mouth and then send those bacteria hopefully to the stomach to be destroyed before they cause any type of infection in the body. Then we have IgA. These are immune proteins. Called antibodies. Specific to one foreign body. So your body, when you are, for instance, encountering the flu virus, will produce antibodies. These proteins that specifically bind to this unique flu that you happen to have accidentally encountered. And you will keep copies of that kind of protein, that kind of antibody for your whole lifespan. IgAs are specifically antibodies that are found in fluids in the body. And saliva, being a body fluid, contains these protective proteins so that if you encounter that flu virus that you had 5 years ago, hopefully this IgA would bind to that flu virus and destroy it before it got in your body and caused any signs or symptoms. So IgAs are awesome. And last but not least, we have mucin. Mucin, as we said earlier, from the mucus cells, mixes with water to form mucus, and the purpose of the mucus in the saliva is to join food particles for ease of swallowing. To form the bolus. So that solid piece of food that we'll easily be able to swallow. Now, when we're looking at saliva is something that is produced all day, every day. So we have what's called intrinsic production of saliva. This is constant saliva production. It occurs throughout the day, all day, keeps the mouth moist, continues to move saliva down to the stomach. If anything were to get into the mouth that could possibly cause an infection of any kind, a bacterium, a virus, whatever that case may be. The salivary swallowing or the saliva buildup triggers the swallowing reflex, and that then, of course, would allow stomach acid to help us get rid of any foreign bodies. We also have extrinsic production, and this is due to the sight, smell, or thought of food. And when that happens, our salivary glands make extra saliva so that we have plenty in case we have the opportunity to take in food. So we'll do one last thing, and that is the process called, just a term, mastication. Mastication is chewing. This is the breakdown of food primarily by the teeth and the assistance of the tongue to position the food properly at the teeth. Also, utilization of the tongue rubbing food across the hard palate, which also would help to break it down. And the mixing of that food, those little pieces that we create with saliva, to form the bolus, the solid piece that we swallow. So the mastication is actually the chewing component, the breaking of food into smaller little pieces. So next, we're going to find out where those small little pieces of food go when they leave the mouth, and that's going to be the pharynx. Our salivary glands include 3 structures surrounding the oral cavity. The salivary glands include the larger parotid gland that we see here in front of the ear that has ducts or tubes that empty into the mouth, as well as our submandibular gland and our below the tongue sublingual gland. And each of these little glands, we'll notice, has tiny little ducts that lead into the oral cavity or the mouth. So the salivary gland is composed of 2 major cell types. We have the serous cells and we have the mucus cells. Serous cells secrete enzymes in a watery fluid. Mucus cells secrete, as they have from the time we first introduced them, as part of columnar epithelium, they secrete the protein mucin. And mucin is going to combine with water to, of course, make mucus for us, which will help to stick food particles together to form our solid piece that's easily swallowed called the bolus. Now, these salivary glands very obviously produce saliva, which has several different roles in the mouth. One thing that saliva does is it helps to dissolve and moisten food. That's due to the water content of saliva. Saliva also begins process of starch breakdown. So we've got carbohydrate breakdown beginning in the mouth. And this is due to an enzyme called salivary amylase. The primary enzyme that the serous cells produce as part of saliva. So amylase is the enzyme that breaks down starch. ASE ending, it's an enzyme. The other thing saliva does with its water content is it helps to cleanse the mouth. So it's going to take any foreign bodies out of the mouth, moves them towards the pharynx and the esophagus and ultimately the stomach to be broken down and destroyed by stomach acid. So this little diagram at the bottom of the picture we were looking at earlier does a nice job of showing the serous cells as well as the mucus cells of the salivary glands. And we also have a look at those mucus cells, those nice, large, plump cells that help to make mucin. So if we look at the composition of saliva, what's it actually made of? Well, it's mostly water. That's going to help deliver the components of saliva to their destination. It also contains ions or charged particles, so there would be some sodium there, there would be some chloride there, there would be some potassium there, amongst other ions. And then we have the main components of saliva that are unique to this particular fluid in the body. So the first is that specific salivary amylase. And as we said a moment ago, its job as an enzyme is to break down starch. So the main job of the mouth chemically is to break down starch. It begins starch breakdown for the body. Another enzyme is present in saliva, though it really doesn't do anything. It's called lingual lipase. It's an enzyme supposedly for lipid breakdown, but it is inactive unless it's in an acidic environment, which would mean in the case of our digestive system, this enzyme doesn't do anything until it reaches the stomach. But it has a little problem. For a lipid to be broken down, an enzyme must do the breakdown. But enzymes are water-based, and lipids are fats. And we all know that fats and water don't mix. So lingual lipase, even if it's activated by stomach acid, does not have the ability to bind to a lipid. So lingual lipase does not do anything in our body appreciably for breaking down lipids. We'll find as we move into our small intestine that there is a chemical that the liver produces that allows enzymes to bind to lipids to break them down, and that substance is called bile. And the reason bile works is that bile has two parts. It has a part that binds to the watery-based enzyme, and it has a second part that binds to the lipid or the fat-based fat that we want to break down. And I have to have that intermediary. So there are other lipases. We'll find there's a pancreatic lipase, and it will actually really break down fats later on in the digestive tract when we get down into the intestine. So lingual lipase is a bit of a dud. It really doesn't do anything even once it's activated by stomach acid. Another component of saliva is lysozyme. This is an enzyme that breaks down bacteria. And the goal of lysozyme is to remove bacteria from foodstuffs. So any bacteria that we consume will be directed towards the stomach as part of the digestive tract and would be exposed to stomach acid. But if we were to happen to ingest a large component of bacteria, a large what we call bolus, a big amount, then lysozyme can reduce the amount of bacteria before the bacteria really enter into the body very far. So we'll reduce bacterial numbers at the mouth and then send those bacteria hopefully to the stomach to be destroyed before they cause any type of infection in the body. Then we have IgA. These are immune proteins. Called antibodies. Specific to one foreign body. So your body, when you are, for instance, encountering the flu virus, will produce antibodies. These proteins that specifically bind to this unique flu that you happen to have accidentally encountered. And you will keep copies of that kind of protein, that kind of antibody for your whole lifespan. IgAs are specifically antibodies that are found in fluids in the body. And saliva, being a body fluid, contains these protective proteins so that if you encounter that flu virus that you had 5 years ago, hopefully this IgA would bind to that flu virus and destroy it before it got in your body and caused any signs or symptoms. So IgAs are awesome. And last but not least, we have mucin. Mucin, as we said earlier, from the mucus cells, mixes with water to form mucus. And the purpose of the mucus in the saliva is to join food particles for ease of swallowing. To form the bolus. So that solid piece of food that we'll easily be able to swallow. Now, when we're looking at saliva, saliva is something that is produced all day, every day. So we have what's called intrinsic production of saliva. This is constant saliva production. It occurs throughout the day, all day, keeps the mouth moist, continues to move saliva down to the stomach. If anything were to get into the mouth that could possibly cause an infection of any kind, a bacterium, a virus, whatever that case may be. The salivary swallowing or the saliva buildup triggers the swallowing reflex, and that then, of course, would allow stomach acid to help us get rid of any foreign bodies. We also have extrinsic production, and this is due to the sight, smell, or thought of food. And when that happens, our salivary glands make extra saliva so that we have plenty in case we have the opportunity to take in food. So we'll do one last thing, and that is the process called, just a term, mastication. Mastication is chewing. This is the breakdown of food primarily by the teeth and the assistance of the tongue to position the food properly at the teeth. Also, utilization of the tongue rubbing food across the hard palate, which also would help to break it down. And the mixing of that food, those little pieces that we create with saliva, to form the bolus, the solid piece that we swallow. So the mastication is actually the chewing component, the breaking of food into smaller little pieces. So next, we're going to find out where those small little pieces of food go when they leave the mouth, and that's going to be the pharynx.